Hello everyone. In this lecture, we will discuss about the assumption of ANOVA. These are the aims of this lecture. These are the assumptions of the ANOVA. In the linear regression, the assumption is known as homoscedasticity. This assumption must be met because ANOVA is based on the linear additive model. Violation of any of these assumptions affects the sensitivity of the F-test and can result in an invalid conclusion. This assumption is commonly known as the unforgiving assumption. Non-independence of residuals can have a serious effect on the validity of the F-test. This assumption is usually achieved by adequate randomization, and it is assumed that the residual of an observation is not related to that of another. We can plot residual versus order of the data to assess this assumption. If the assumption is met, we expect to see no pattern in the plot. Yes, often the correlation between observation is known and expected. For example, we want to know the effect of nitrogen fertilizer on the total yield of sugarcane calculated from 3 harvest time: plant, ratoon 1, and ratoon 2. This type of data is often called repeated measures or longitudinal data. It is closely related to time series data. This correlation is known as a temporal correlation. Another example, the yield of adjacent plots are likely to be more similar than the yield of plots from a different part of the fields. This correlation is known as a spatial correlation. If possible, we can use an experimental design to account for this correlation. For example, use harvest time as a blocking factor or use randomization to ensure that a particular pair of treatments or the same treatment does not always occur in adjacent plots. Alternatively, we can use the general linear mix model to relax the assumption of independence. The normality assumption is important for the validity of the F-test in ANOVA. Fortunately, the F-test and the t-test are robust tests. They are still approximately valid even when the data are not normal. To avoid serious departure from normality when sampling from plots, we should use sample means based on several within-plot measurements. Remember the central limit theorem? We can use a normal probability plot or the normal QQ plot of the residuals. If this assumption is met, the residual should be a straight line. Yes, there are situations where the measurements are markedly non-normal. For example, counts of insects on plants or number of seeds germinating. If the assumption of normality was invalid, there are three options. Use the non-parametric test that does not require the assumption of normality. Use data transformation techniques so that the data become normally distributed. Or use a generalized linear model. The F-test uses pool residual variance, so if this assumption is not met, it can lead to serious errors. If group sizes are equal, the F-statistic is generally robust to the violation of this assumption. If group sizes are vastly different, the F-statistic will be biased. In most experiments where the difference in treatment effects are small, this assumption is usually met. We can use residual versus fitted value plot to assess this assumption. If this assumption is met, we expect to see no pattern in the plot and similar spread of residuals across all fitted values. Often, this non-homogeneity of variance is due to non-additivity of the treatment effects. Therefore, a data transformation that corrects for one corrects for another. Alternatively, we can account this non-homogeneity in the general linear mixed model. The effect of non-additivity is similar to the effect of non-homogeneity of variance. ANOVA is based on the linear additive model. The analysis assumes that the measurements are made of components which are added together. Additivity means that each treatment contributes equally to the response variables. When the data are not additive, treatments are not contributes equally to the response variables. We can use data transformation. Alternatively, we can use nonlinear model. Data transformation is an option that may allow the assumptions to be sufficiently satisfied. These are some commonly used data transformation. The log transformation is often required for counts, such as the number of disease plants per plot and number of insects per plant. Data require a square root transformation are typically the number of events per unit of time or space when the event is considered rare. The arcsign transformation is suitable for percentages based on counts. In general, data transformation should be avoided because of these two reasons. One, a transformation that corrects for one violation of assumption may introduce another. And two, it is often difficult to interpret the result of the analysis using transform data. Before deciding to use data transformation, we should always use the residual plots to check the assumptions. The violation of the assumption could also occur due to outliers. Rather than using data transformation, we can use other alternative methods. In non-parametric methods, no assumptions are made about specific population distribution or their parameters. These methods are usually used for non-normal data. However, not all non-normal data can be analyzed using non-parametric methods. These methods also have assumptions that must be met. A generalized linear model is suitable if the residuals follow one of a number of distributions other than a normal distribution. We have to know what is this expected distribution. A general linear mixed model assumes normality, but it can account for the non-independency and non-homogeneity of variance. Now, can you do this?